Hello, everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia, and I'll be your host for today's session. Myself and our wonderful team of co-organisers, Andreas, Ulia and Wouter, in the groups of Judith Mank and Simone Imler at the Universities of British Columbia and of East Anglia, are very grateful for the enthusiasm from the research community for this initiative, and we've been honoured to host such a diversity of great minds so far. So thank you all for your fantastic engagement in the series. Before I introduce today's speaker, I'd like to remind you that there will be a Q&A session directly after the talk, so please post your questions in the dedicated Slack channel and upvote questions you'd like to hear. We aim to keep the whole session to 1 hour in length. So it is my great pleasure to introduce today's speaker, Professor Rosemary Grant, Emeritus Professor of Evolutionary Biology at Princeton University in the USA. Rosemary's research explores how new species arise and the evolutionary and ecological drivers of phenotypic and genetic variation. During a remarkable 40-year career studying adaptive radiation in Darwin's finches in the Galapagos archipelago, together with her husband, the fellow renowned evolutionary biologist, Professor Peter Grant, their intensive and meticulous work has elegantly demonstrated evolution in action, and that natural selection on beak and body morphology can respond rapidly to severe drought and changes in food availability. More recently, Rosemary's work has focused on the genetic and genomic underpinnings of this trait diversification and the barriers to hybridization. Rosemary has received many prestigious awards throughout her career. These include the E.O. Wilson Prize, the 2002 Darwin Medal from the Royal Society, before being elected as a Fellow of the Royal Society in 2007, and being awarded with their Royal Medal in 2017, in addition to the coveted Darwin Wallace Medal of the Linnean Society, to name just a few. This is testament to Rosemary's outstanding contribution to the study of evolution, biodiversity, and conservation biology. Today, Rosemary will present her talk entitled Darwin's Question Revisited, How and Why Species Multiply. So Rosemary, thank you for accepting our invitation, and over to you.
Well, thank you very much for that very kind introduction, and I will go straight in. I have put Peter's name also on my first slide because we have done this work very much together. So Darwin's poetic last sentence of his Origin of Species reads, "There is grandeur in this view of life. Whilst this planet has gone cycling on according to the fixed laws of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved." And indeed, this Nassau image captures the magic, the uniqueness, and the fragility of our planet, with the biodiversity being its fundamental feature. But how do we study biodiversity? How and why do species multiply? So how do we even go about beginning to study speciation? Well, Norty and Touts have put this very succinctly. To understand the mechanism of speciation, the focus should be on cases of incipient speciation rather than on completed ones. And indeed, we know that much diversity arises from adaptive radiations, that is, the rapid diversification of lineages from a common ancestor, such as these Hawaiian honey creepers, which have evolved into these wonderful forms, different colors, different sizes, and extremely different beak shapes. But unfortunately, all these species, or many of these species, have gone extinct, and also it is an old radiation. I'll just stop here to say, can you see my whole slide? Because I can't. You can, okay. So what we really need is a young adaptive radiation, and what would be even better is a young adaptive radiation on an island archipelago. And this is because islands are likely to differ from each other ecologically and impose different selection pressures through both space and time. And this gives us an opportunity to measure changes that are still ongoing in contemporary time. So this is the reason we chose initially Darwin's finches on the Galapagos. We thought it was a young radiation at the beginning, and then genetic work has shown that indeed it is a young radiation. The whole radiation of 17 species has arrived in the last 1 million to 2 million years. Furthermore, these species occur together and separately on the different islands. Most of the islands are completely uninhabited, so they're close to being in pristine condition. So we know that any changes we measure will be the result of natural causes and not the result of human interference. And furthermore, it's a dynamic environment, and this is very important. So it sits astride the equator, and so here's the archipelago. And as such, it is subject to the El Nino Southern Oscillation phenomenon. So in El Nino years, which occur about once or twice a decade, the trade winds cease, the water around the islands becomes very hot. This brings torrential rain to the islands. The vegetation explodes, lots of food for the finches, and the finches breed. In other years, also occurring once or twice a decade, there are what they call La Nina years. And here the trade winds continue to blow, the waters around the island continue to be cold, no rain falls, there are droughts on the island, and we now know that on the little island that we study of Daphne, that as many as 80 to 90% of the finches die. So we have gone to all the islands in the archipelago. We have done short research on these islands and collected blood samples, but we did in-depth research on 2 of them. One is Genovesa in the northwest, and the other is the little island of Daphne Major in the center of the archipelago. And it's the work on Daphne that I'm going to talk about today. Excuse me, when we first went to Daphne, there were 2 species of finches, and large number. The reason why we chose the island is that there were large numbers of these 2 species of finches. One of them is Fortis, the ground finch, and the numbers we now know in El Nino years will extend up to over 1,000 individuals, and during droughts will drop down to about 100. In Scandens, the range of numbers goes from 700 to 800 and then down to less than 100. Now, we knew from David Lack's work that Fortis was exceptionally variable in body and beak size. And if you translate this into human terms, it will be the equivalent to a 7-foot adult and a 3-foot adult human. So enormously variable. The island is small. It's 3/4 of a kilometer in diameter, 120 meters high. The finches are resident, and unlike on the larger islands, they cannot escape droughts by going up to higher elevations. So they're captured on Daphne. And now this is the outline of my talk. I'm going to talk about Darwin's model of speciation, first of all, and the evidence we have. We now call it allopatric speciation with character displacement. And then I'm going to look at the causes and evolutionary consequences of very rare episodic gene flow between the species. And all this will give us insight into 3 different models of speciation, and that's how I'll end my talk. So I'll introduce you to the finches first of all. This is Fortis, the medium ground finch. This is the finch that was very variable, that Lack pointed out was so variable. And so the first thing we did when we went to the islands is caught a lot of these finches. We gave them unique combinations of color bands, and then we watched what they were feeding on. And we found that the small birds, and here's a picture of one of the small finches up here, they were eating solely small soft seeds, whereas the large finch, and can you see this? Because on my slides you can't. Oh, yeah, you can. Okay, so this is a member of the large population, and we found that what they were feeding on were the large tribulus beads and the large opuntia beads. They were also taking a few small and medium-sized seeds, and the medium-sized birds were taking the medium and small seeds. Then we have Scandens, the cactus finch. It's the only species with a really strong pointed beak, which can open the opuntia fruits and get out the seeds. Now, it makes a large hole in the opuntia fruits, and other birds will use that hole, but Scandens is the only one that can actually open them. And then when the opuntia flowers are in bloom, the Scandens is feeding almost exclusively on opuntia pollen and nectar of opuntia flowers. And the blooming of opuntia flowers lasts for about 4 months. So this is Daphne. You can see that it's all slopes. It is an uninhabited island, not too surprising. There's no water on it. And it has a large crater in the middle. On the walls of the crater, you see the green plants moving up there are opuntia bushes where the birds breed, and they also breed in the opuntia bushes on the outer slopes. The birds go everywhere, and so do we. And you can also see from this slide, it's not the easiest island to get on. This is our landing, the easiest point of the island to get on. These white arrows are not painted on the rocks. I've just put them in on the photograph. But we quarantine everything, our food and our equipment. And once we've taken up all our water, there's not even a drop of water on the island. We have to take all the water with us, all our food with us, all our equipment. We stay there. And we stay there for, Peter and I have stayed there for 3 or 4 months. And we've also had students there for quite long periods of time. This is our bedroom. This is our kitchen in a shady cave. We get up very early in the morning. We put up nets. We catch the birds before the sun strikes the net. And then we take the birds into a cave. We weigh them, measure them, band them, and take a small drop of blood for later DNA analysis. Then the rest of the day is looking for nests, and we record who's pairing with who, the number of eggs they lay, the number of fledglings. We record their song, we record what they're feeding on, and we record the amount of food resources on the island at that time. So Darwin's model of speciation, a hypothetical model of speciation, can be thought of in 3 steps. The first step, he envisaged the birds came from the adjacent mainland, landed on an island, met very different ecological conditions through the process of natural selection, and over the generations, they gradually adapted to these different ecological conditions. Population built up, moved to another island. Step 1 and step 2 would be repeated over and over again, until step 3, when they came back into secondary contact. And at this time, he envisaged there would be competition for resources and further divergence, what we now call character displacement. And I'll be telling you, giving an example of this later. Over the years, Fortis, remember this is a very variable finch, has changed very much so. So in 1973, when we first arrived on the island, the average beak depth, this would look very similar if I'd used body size, but I'll just use beak depth here. The average beak depth was here, where's my pointer? Here. And then these tram lines on either side are the 95 confidence limits on either side of the 1973 sample. And you can see that there's one period where the average beak size increased enormously, stayed this way for a while, dropped down, and then there was a precipitous drop here in 2003 to 2005. Now, there's one common denominator of all this, and this was a drought. Every single one of these changes involved a drought. This is what the island looks like in a normal year. This is the same picture in a drought year, and you can imagine that the finches are scrabbling about in the rocks, looking for the last few remaining seeds. And this is when we get a lot of mortality. So we know from Haldane that if we are going to measure evolution by natural selection, there has to be 3 steps. First of all, we must know that the traits that we're interested in are heritable. And our traits were beak size and shape. So we took 3 measurements of beaks, and body size. And we took 3 measurements of body size. And as a result of some environmental perturbation, there is differential survival. And in our case, the environmental perturbation were droughts. And then in the next generation, the offspring of the survivors must resemble their parents in the trait of interest. And of course, this would happen if the traits are heritable. And if the traits were not heritable, then we would just go back to the starting point. So the first thing we did was we banded a lot of birds. We followed them and their offspring. We took the adult mid-offspring and we looked at the association between adult mid-offspring and mid-parent. And we found that the heritability was extremely high. The heritability of 0 is no heritability. Heritability of 1 is absolute 100% heritability. But ours were all our measurements of the 3 measurements we took were always between 0.7 and just above 0.8. So they're all very high. And we did this heritability studies at different times throughout our study periods. Well, we didn't have to wait long for the first drought. It occurred in 1977 when no rain fell that year at all and finches began to die. What happened was the small soft seeds went out first, leaving these large, hard, tribulus seeds, and it's only the large fortis that were able to crack these large, hard, tribulus seeds. So as the months went by, the birds died, but the small beak birds died disproportionately, leaving few birds, but they were mainly the large birds that could crack the large, hard tribulus seeds. Because of the high heritability, when the rains came back and these large birds bred with each other, then there was an evolutionary response to this. This is the average, this was the average size of the offspring. So there was an evolutionary response to a natural selection event, which occurred here. And you can see that the average size of the beak size went shooting up. And it stayed that way for a while, came down a bit. Then we had this precipitous drop here. Now, this was interesting because this was a 2.5-year drought. Can you imagine? No rain fell for 2.5 years. And the 90% of the birds died. Now, exactly the same happened. The small seeds went out first, leaving the large, hard tribulus seeds. So we would have expected that fortis would have been selected to go up, just like it did in 1977. But we have this precipitous drop. Now, why is this? Well, the reason is that this bird, Magnorostris, had arrived on the island in 1983. There were 5 birds, and they stayed to breed, and the population expanded to, by the time this 2.5-year drought, which started in 2003, by that time in 2003, there were well over 250 individuals on the island. Now, Magnorostris loves tribulus. It's a bird twice the size of a fortis and very aggressive. So you can imagine that what happened is that Magnorostris outcompeted the large fortis for the few remaining tribulus seeds. And it was interesting, the small birds were dying because there were very, very few small seeds, but they actually changed their behavior. And what happened is when Magnorostris cracked these tribulus seeds, out of these little locules scattered some very small seeds. And so the small birds ran up followed the Magnorostris around the island. As the Magnorostris cracked the seeds, they would go in and pick up the small seeds. And I have a video of them doing this. And here it is. So there's a Magnerostrus cracking a seed. A little tortoise goes up and grabs one of the seeds. And you'll see it again at a slightly slower speed. So when these few surviving small birds, when the rains came back after 2.5 years, they bred with each other, produced small-billed offspring because of the higher heritability, and the average size of the population plummeted. So this is just exactly what Darwin envisaged, that there'd be a character displacement event as these birds came back together again. But we could ask, are there any specific alleles that underlie these beak size changes? And initially, we thought the response would be solely polygenic, involving many loci, each with very small effect. But our collaborator, Leif Anderson, who's here with his lab group, and the next work I'm going to talk about is the genetic work done by his graduate student, his PhD student at this time, who is Sangeet Lamijeni, and he's now a professor. So Leif thought, well, it's really worth looking at all your blood samples to see if there was anything. And so he took all our blood samples, not just from Daphne, but from all the other birds. And he matched up large with small, large and medium with small, and came up with a 525 kilobase region, which contained the gene HMGA2. And he knew about this as being important in size in mammals. And what was really exciting is that he found that there was an allele with HMGA2, which is associated with large size and one with small size. And then they were segregating in our tortoise population. So what we did is we sent 70 samples over to Leif, blood samples from birds where we knew the measurements, but Leif and his lab group did not. And they sent back the results. Homozygous HMGA2 large were indeed large build. Let's see if I can get my, here we are, large build. And homozygous HMGA2 small were small, and the heterozygous were intermediate. So we then asked, well, how did they survive during the drought? So out of the 20 homozygous HMGA2 large, 14 died through the drought. And out of the 19 for small, only 5 died through the drought. So initially, we thought this response, as you say, would be solely polygenic. But here we could see that a single loci facilitated a rapid diversification together with some polygenic variation. And the selection coefficient was a whopping 0.59. Which is huge when you think it is just on one loci. So that was a very exciting result. And so in conclusion for this bit, we have a natural selection with an evolutionary response. We know that it occurs when the environment changes, and during droughts. We know why we get this response, because it was the tribulus seeds that they were eating, and it was only the large birds that could do this initially. And then we know that other species like Magnerostrus can determine the direction of evolution, as we've just seen it do in the character displacement event with tortoise. So now I'm going to move to the next part of my talk, which is the causes and evolutionary consequences of rare episodic gene flow between species. And part of this hybridization will lead me to the formation of a new lineage, which we have followed from its inception for over 6 generations. But our definition of species, I'm always asked about this, but our definition of species is that species are a reproductively cohesive group of organisms that are identifiably distinct, just like these on Darwin's finches. The reproductive barriers between them are rarely or never penetrated. Now, we know that gene flow, I'm going to try and get rid of this, that gene flow between species has been found in numerous instances across all forms of life, from bacteria to our own ancestors. We've only got to think of the wonderful work started by Svante Pääbo, which shows that there was hybridization between our own early ancestors and Denisovans and Neanderthals. But it has also been shown to be a source of variation used in adaptation to new environments. And a good example, many examples, but this is a nice one from last year, where toxic butterflies have acquired genes for warning coloration from other species. Oops. Oh, by moving this, I think I have stuck my, now it's come back again. My PowerPoint went stopped. So we had many cases where we have full genome analysis have allowed the detection of hybrids, but very little is known about the initial stages of hybridization that could eventually lead to speciation in nature. And it is this that I'm going to talk about today. It's about the initial stages of hybridization that can eventually lead to speciation. But first of all, we have to have two major questions. What are the reproductive barriers? How are they formed and how are they breached? And what are the consequences of this introgression? Is it a source of variation used in adaptation to new environments? And could it lead to genetic incompatibility? So first of all, what are the barriers to reproduction? How are they formed and how are they breached? Well, with Darwin's ground finches, we can't use plumage because all males are black and females, like this black Magnorostris, and females are brown, like this female Portus. So they're similar in plumage, they're similar in type of nest, and they're similar in courtship behavior. They differ markedly in body size. This is a 32-gram Magnorostris compared with a 12-gram Polyginosa. They differ in beak shape, comparing this massive bill of Magnorostris with a sharp pointed bill of Scandens, for example. And they also differ in song. And here are sonograms showing the Magnorostris song, Portus song, Polyginosa song, and Scandens song, all very different from each other. So we recognize the birds by their morphology, beak dimensions, body size, and song, but we had to know if the birds could do this. So initially, when we first arrived on the islands, we did mount and playback experiments to ask if individuals can discriminate between their own and another species. First of all, with morphology alone in the absence of any song cues. And to do this, we used stuffed museum specimens, a female Portus on one end, female Scandens on another, took it into the territory and asked the living territory owner, can you discriminate your own female from a female of another species? We did this many times, and the answer was a resounding yes. We then used playback to ask if they could discriminate between song in the absence of any morphological cues, and again, the answer was yes. So we know that the barrier to reproduction is song and morphology, and the reason, and we know some other things, we know that finches choose their mates on the basis of morphology and song, learned very early in life during a very short sensitive period. This period was worked out by Bowman early on, and it's between day 10 and day 40 after hatching. And this coincides with the last few days in the nest and when the birds are out of the nest being fed by their parents. And all this time, the father is singing. So it's not too surprising that the young males grow up singing perfect renderings of their father's song. And the females, although they don't sing, recognize members of their own species through the song and morphology, which is learnt in this period. This is an example. It excuse me, doesn't occur in all birds, but it occurs in many birds. And this was first worked out by Konrad Lorenz and is termed sexual imprinting. With Lorenz, he first worked it out with goslings, with geese, where he took the mother away. The goslings first saw him, imprinted on him. And so when they grew up, they tried to mate with his boots. So in Darwin's finches, the pre-mating barrier is based on song and morphology learned early in life. But it's vulnerable because it can be breached if a young bird hears and learns the song of another species during this short sensitive period. This happens extremely rarely, but it does happen, and it can happen if the father dies and the mother is left to rear the offspring. And if the natal neighbor is a bird of another species, then it will learn that species' song. Now, Magnorostris that colonized the island, as I've told you, gave us an opportunity to test this. I remind you that Magnorostris is twice the size of Fortis, almost twice the size of Scandens, and is very aggressive. It has a very loud song. Here's a Magnorostris song down here. And over the years, we had 8 Fortis and 2 Scandens that learned and sang a Magnorostris song. So here's one of the Fortis singing a Magnorostris song. This is a normal Fortis song here. One of the Scandens singing a Magnorostris song, normal Scandens song here, and none bred with the Magnorostris. And the reason was when these little birds grew up, opened their mouths and sang a Magnorostris song, a Magnorostris would whip in as if from nowhere and beat them up. So it never led anywhere. So it seems that size really matters. The barrier is when the size difference is large, as it is between Fortis and Magnorostris, the barrier is robust. Even learning another species' song did not lead to hybridization. But when the size difference between species is small, as it is between Scandens and Fortis, and even though Scandens and Fortis are not each other's closest neighbor, learning another species' song can lead to hybridization. And again, this happens rarely. It happens again about 1% of the birds. So we had this hybridization between Scandens and Fortis, and there's also something I haven't mentioned up until now, and that's Polyginosa, which is the very smallest ground finch, and that is an occasional rare immigrant to the island. And 1 or 2 come every few years or so. It's not regular, but when it comes, and the ones that do breed, they all breed with Fortis. So there are very few of these, but it leads to an interesting problem. It means that there's hybridization between Polyginosa and Fortis, and we know this hybridization rarely between Fortis and Scandens. Everything is very rare. But Fortis acts as a conduit for the passage of genes from Polyginosa to Scandens that themselves never hybridize because the size between Polyginosa and Scandens seems to be too great. And we have never observed Polyginosa hybridizing with Scandens on Daphne. So we are actively pursuing this now, and we're pursuing it genetically, so stay tuned. But I'm going to concentrate mostly from now on between the very few hybrids that were formed between Fortis and Scandens. Now, none of these hybrids of any kind actually survived the dry season in the first 10 years of our study. The dry season was very dry for all these 10 years. Initially, we thought maybe we were seeing genetic incompatibility, but we also thought, well, a lot of the birds are dying, it's very dry, and there may not be the appropriate food. And this seems to be right, because in 1983, we had an immense El Nino year. It was, from coral core data, the strongest in 400 years. And rain fell for 8 months solidly of the year, and birds bred for 8 months, once a month. So birds were breeding 8 times that year. So this is what a normal wet season looks like. And the plants in the foreground, the low-lying plants in the foreground you see are tribulus plants. And they produce these large hard seeds that made the difference between survival and non-survival during the drought. But in this year, rains came and grasses and small seeded plants grew up, smothered the tribulus. Vines grew over cactus bushes, vines grew over trees, and even in the year following, the island was draped with dead and dying vines. And our quadrats, where we measure the seed production, showed that the island had been converted from a large hard seed producer to a small soft seed producer. And under these circumstances, the hybrids began to survive. But we could still ask, how well did they survive compared with the parental species? And so these are 3 years when a large amount of rain fell immediately after this. And we compared the survival of hybrids in solid circles, in black solid circles, with the survival of the pure species, Fortis and Scandens, in open circles. And in this altered environment with an abundance of small soft seeds, the hybrids survived as well as, if not better than, the pure species. They produced as many offspring, they produced as many fledglings and as many recruits as the pure species. So we had no evidence of genetic incompatibility under these changed environmental conditions. Now, the hybrids didn't breed with each other, but they followed the rule. You breed according to the song you learn early in life. And so they backcrossed according to song. And so we had gene flow going from Fortis to Scandens and Scandens to Fortis. Now, initially, it was Fortis, Scandens into Fortis. And we got the change in beak shape in Fortis. Fortis became much more pointed and much better adapted at dealing with small seeds. And these pointed bills remained for the rest of for the next 30 years in our study. After 1990, this became extremely interesting because the introgression was now mainly from Fortis into Scandens, and it was mainly from female Fortis into Scandens. So what happened is that Scandens bills became blunter and blunter on average and much more Fortis-like from 1990 onwards. Now, at this time, we just had microsatellite, but there was enough private microsatellites for us to be able to track genes flowing from Fortis to Scandens and Scandens to Fortis. And we found that they converged on each other genetically, and also on the same trajectory, they were converging morphologically, which we could tell from our measurements. But again, we went to Leif, his lab group, and asked, are there specific alleles that could have contributed to changes in beak shape? And he took again all our blood samples, and again he matched all the blunt beak species with the pointed beak species, and he found an area, ALX1, a gene that encodes a transcription factor, which he knew was very important in craniofacial development of mammals and some birds and fish. In fact, a mutation in ALX1 contributes to cleft palate in humans. But for us, what was again very exciting was that there was an allele for pointed beaks and allele for blunt beaks segregating in the populations on Daphne. So as I said, the introgression was from Fortis into Scandens, but very importantly, the introgression was only from, or mainly from, female Fortis into male Scandens at this point. And this, and also when we got, because the sex ratio, this occurred, this skewness occurred, being only females going into Scandens, was the result of a very skewed sex ratio in Scandens. It was skewed in favor of males. And in fact, during this period, there were even some years when there were 100 male Scandens to only 16 female Fortis. So you can imagine that it was the Scandens males, they didn't have enough females of their own, so they would hybridize or try to hybridize with the Fortis females. And even amongst the hybrids, the Fortis Scandens hybrids, it was the Fortis Scandens females that mated with the Scandens males. because the Scandens males are larger than Fortis scandens males, and they outcompeted them for territories and for females. So we had this situation which we had followed, tracked with individuals. So, and just to give you an idea of after, excuse me, after hybridization had been going on for a while, these 3 photographs were taken from Scandens, which came out of the same net in 2012. We have some Scandens up at the top that have the long pointed bills that have identical measurements to Scandens before hybridization started. And then there are others with really blunt bills like this, which are much more Fortis-like, and they are the result of Fortis genes penetrating the Scandens. So what Leif did and his group, they took, made 6 groups from our blood samples. They took Fortis and Scandens, which they called early. That was Fortis and Scandens blood samples before hybridization. And then they took, after hybridization, they divided Fortis into those that were late pointed and late blunt, late meaning after hybridization, and the same with Scandens, late pointed and late blunt. And they did pool whole genome sequences for each of these 6 pools of 30 birds each. And then they did, neighbor joining phylogenetic tree based on the SNP data. And what came out with the autosomes and the mitochondria, that there was a major shift of the Scandens blunt-beaked birds that had shifted towards Fortis, both in autosomes and mitochondria. And remember that it was a female Fortis and female hybrids that were going into the Scandens population. So this exactly followed what we expected from our field observations. And then when we looked at the, somehow I have seized up again. Okay, thank you. So then when we looked at the Z or Z chromosome, there was reduced gene flow on the Z chromosome, as we would expect, knowing that it was the female Fortis, female hybrids that went into Scandens. And so this was exactly what we had expected. But the reduced gene flow on Z has been found in many other ZW, ZZ systems where females are the heterogametic sex. And it has always been explained by the large Z effect and the faster Z evolution contributing to genetic incompatibility. Well, we know in this case it came about through social incompatibility. So this raises the question, could social incompatibility, might it possibly be a first step in the evolution of genetic incompatibility? That would be quite an exciting finding, and we are looking into this genetically at the moment. So now I'm going to move on to the new lineage, the Big Bird lineage. In 1981, Trevor Price, who is now a professor at Chicago, he was a graduate student on the island, and he had all the birds on the island banded. And then suddenly a bird arrived on the island. It was a young bird, and it looked superficially like Fortis, but it was massive. Fortis is 17 grams. This one is 28.5 grams. When it was a young bird when it arrived, when it sang, it had a completely unique song, never heard on Daphne before, never heard anywhere on the island. And he banded it 5110. We came onto the island later, caught it again, and this time we took a small drop of blood. So we have the blood sample from him. And we had, both Peter and I had written in our notebooks, this looks awfully like a Conirostris from Hispaniola, but can't possibly be because it's so, Hispaniola here is so far away. But Leif and his group took the blood sample, and sure enough, it was a Conirostris from Espanola. So it took ages to breed. It had a beautiful territory. It was a large bird, much larger than Fortis, but it couldn't get a mate. After all, it was the wrong size, and it sang the wrong song, and it just sat there on this beautiful territory. And then eventually, at the very end of one breeding season, a female Fortis lost her mate, and here was this beautiful territory, so she paired up with this bird, and they produced offspring. And then he paired up with another Fortis as well. So they produced offspring. We got blood samples from them all, and they had showed genetic transmission from 5110, and all the males sang 5110's weird song. And then along came the 2.5-year drought, and all these birds went out except for these two, an inbred brother and sister. When the rains came back 2.5 years later, the brother and sister bred with each other and produced 26 offspring, all but 9 of which survived to breed. And we had a daughter breeding with her father, a son breeding with a mother, and the rest of the sibs breeding with each other to produce more sibs which bred with each other to produce more. All these birds showed genetic transmission from 5110, all sang 5110's song, and all were large like 5110. The genome-wide inbreeding coefficient marched steadily upwards. It got to 0.6 over the 6 generations. But interestingly, there was an allometric shift. The red dots are Conirostris from Espanola, the green ones Fortis from Daphne, and the blue ones are this, what we now call this Big Bird lineage. And you can see that it's displaced and that they have a big head and a big bill on a small body. Now, we measured how efficient they were dealing with tribulus, and we found that they were just as efficient as Magnorostris. You can do this by measuring the cracking time, cracking these large, hard tribulus cases. And so if there was another drought and tribulus was the seed that was left longest, then the big birds would be at a huge advantage, because not only would they be able to crack tribulus as efficiently as Magnorostris, but having a smaller body, they would require less food in order to survive. So this new lineage is behaving like a separate species. It is large, much larger than Fortis, its nearest relative. In morphological space, it lies here between Magnorostris and Fortis, and this is the gap that it was expanded after Magnorostris had outcompeted the large Fortis, which lay up here in the character displacement event, and the big bird population moved right in. It sings a different song. This is 3 generations of the big bird song, very different from Fortis, Scandens, and Magnorostris song. It holds contiguous territories. It has large territories in one part of the island. The blue dots are territories before the drought, the red dots after the drought. And these territories overlap with Fortis, Scandens, and Magnorostris territories, who ignore them completely, and they are ignored by them. So in all respects, the new lineage is functioning as a separate species. Will it die out through inbreeding depression? It might, but it was still alive last year. Will the genetic variation be augmented through gene exchange? It might, but we see no sign of that yet. So whether it survives or not, this lineage gives us insight into how new species could arise and either persist or become extinct. So we started in 1973 with 2 species on the island, Fortis and Scandens. Then we had this enormous El Nino event in 1983. The hybrid survived, and Fortis and Scandens began to merge. Magnorostris arrived on the islands, outcompeted the large Fortis, and the big bird lineage moved right in. So we have 3, this has given us insight into 3 models of speciation. Darwin's model, which was the allopatric model. We had no breeding on secondary contact, and the character displacement led to divergence. Then it gives us insight into fusion speciation, which is the interbreeding of two non-sister species, Fortis and Scandens, with a progression towards complete fusion into a single breeding population, which is acquiring new combinations of genes, and it's phenotypically changed and diverged very much from Fortis and Scandens populations inhabiting other islands in the archipelago. And finally, what I've just told you is the formation of the Big Bird lineage. So this we think of hybrid diploid speciation. So we thank all our students, collaborators, we couldn't have done this work without them, but I just want to leave you with one message. These endless forms that Darwin talked about, the endless forms, most beautiful and most wonderful, we are losing them at an unprecedented rate. The global diversity is being degraded, and we have seen that both species and environments are dynamic, they're constantly changing. And my message that I want to leave you with is to maintain biodiversity and the potential for future speciation. We must keep both environments and populations capable of further natural, and I emphasize natural, change. We don't have much time. Thank you very much.
Thank you very much, Rosemary. That was such a fascinating and interesting talk, and a very interesting story throughout. And yeah, we have a lot of questions coming in, so I'm going to start with some of those.
Okay. Do I see the questions, or you will read them?
I'll read them out to you. So the first question asks whether you could comment or speculate on the extent of recent hybridization of Darwin's finches on other islands, and is the gene flow between islands the exception, would you say?
Well, I think as far as we know, gene flow between islands is probably, it probably occurs, but I think you have to do long-term studies and in-depth studies like we are to catch that. But we do know that, well, we know that polyginosa comes in, but we've also done in-depth studies on Henabasa, and there we see exactly the same with hybridization between the species on Henabasa, so that we can tell. Again, it is a long-term study. And then there has been a study which has been done by other people in the Galapagos, and they have found that there is also hybridization between tree finches as well. So hybridization does occur, and I'm quite certain that there is the occasional flight from one island to another, and where most of the time probably they won't stay to breed, but very occasionally they will stay to breed. So I think we just caught this because of but I don't think it's necessarily the only time it has ever occurred. It can't be. It must occur rarely. It must be ongoing.
Yeah. That's interesting. So you described in your talk about how there's these very strong bottleneck events where you get up to 90% mortality. And so reduced population size. And there's a question which asks, what do you think has been the role of drift in the observed phenotypic variation over time?
I think, well, drift, I could have mentioned this. I mean, drift is occurring all the time. But what we are seeing is, these huge effects of natural selection as well. So both are occurring in time. And you mentioned the bottlenecks that are occurring. These bottlenecks are brief, because as soon as it starts to rain again, what happens is that here the birds have a vast amount of food brought on by the rain, and yet the population is very low, and they explode. So we have not observed much. So, very quickly, and there's a lot of diversity as well. and the diversity is brought in through hybridization. A lot of variation, genetic variation. So I think it quickly goes back. We don't see these huge things that we think that we would expect to see with bottlenecks, because I think it's so brief, starting out with genetic variation. Mm-hmm. And yes, definitely drift is going on the whole time. Yeah.
Great. Thank you. So we have a question about, you mentioned how there is reduced gene flow on the Z chromosome. And so the question asks, are there any indications of Haldane's rule in the heterogametic sex?
No, we found none so far. And we've looked back at this because there are cases where the male hybrids do survive. And so we do know that. But we are actually in the point of looking at this in much greater detail. So at the moment, I will say, no, we have not observed anything yet. But this is ongoing research. And we do know that there are some males that do make it. Yeah.
I see.
But there could easily be the first stages. I mean, it's a really super situation to look at this in more genetic detail, which is exactly what we're doing.
Fantastic. It seems like a very interesting question for sure. So we have a more general question, which asks whether you could comment on the future of long-term studies and what advice can you give to early career researchers on this?
Well, yes, I do. I think these long-term studies are absolutely vital. I mean, you would just think of our study. If it was a 3-year study, which is what most grants are, well, it could have been at any point in the study and we would have missed all the exciting things. So I think there's got to be an in-depth study. The other thing that I could easily have ended this talk with would be saying that, the genomic data we are finding now is enormously exciting. But it doesn't really stand on its own. It is, when it's put together with in-depth field work, then you get the really exciting things coming out of this. And very often there's very little money that is being put into field work, and much more money is being put into the genomic side. I'm not saying I want to cut that down at all, the opposite, but I'd like to see much greater emphasis put on long-term studies combined with genomic data. And so I am constantly trying to encourage long-term studies and also training in the field to get really good data from the field to match the genomic studies.
Fantastic. That's a really interesting perspective. So we'll just end with one final question for now. And it's, I guess, looking more broadly, if you could pick a new system, a new evolutionary question, and devote the next 40 years to it, what would it be?
There are quite a number. I'll go back and say, before I even met Peter and before we decided to work on the Galapagos archipelago, my pet one was so similar to this. My pet one was Icelandic char. And the Icelandic char, we know when the fjords, many of the fjords were closed because from the geological data, the char have diverged into benthic forms, limnetic forms, and many different species. And originally, this was years and years and years ago. In fact, it was in the 1950s. This shows how old I am. So in the 1950s, I had planned a whole project on looking at both the ecology and the genetics, and I'd worked out a little way of how I could do the genetic work on these char in the Icelandic lakes. Then I got married to Peter and we went to the Galapagos. so that's always been, shining there. But there are all sorts of really young adaptive radiations like that. And of course, work has been done on the sticklebacks. It's been done on, lots of work is being done now in all sorts of exciting areas. So, yeah. So that's
fantastic. Yeah, that was really interesting.
So that was that I even got money for it, I think, in 1959. Yeah, but I met Peter in 1960. I think it was 1960 I got money. I met him in September 1960. Yeah.
Fantastic. Maybe something to pursue in the future. Great. So I think that's all we have time for today, but it is our great pleasure to thank Rosemary once again for a truly fascinating and interesting talk. Thank you, Rosemary.
Thank you very much. Thank you.
So our next talk will be from Professor Nicole King at the University of California, Berkeley. So we hope that you can join us on Friday, the same time and same place. And in the meantime, check out our updates on Slack and our Twitter feeds, spread the word about our seminars. And thanks for watching. Stay safe and see you again soon.